Hello everyone. Today we have to discuss on the topic 3D images, negative staining and single particle reconstruction. Myself, Jumana Karadan, 2nd MSc Biology from St. Mary's College, Thrissur, in the guide of Dr. Kain Vadakan from Department of Biology. First, we have to discuss about 3D images. 3D images means three-dimensional images. For the magnified and visual picture of a object in the three-dimensional view, we have to use scanning electron microscope. It works by scanning the surface of an object with a focused beam of electrons with a focused beam of electrons and detecting the electrons that are reflected from and knock off the sample surface. So that is why we get the three-dimensional view of the object. The visual, the visual experience we get from this 3D image is called virtual reality. Three D images which is different from the 2D images. That is, 2D image have the height and the width, but now we get about its depth. But in case of 3D image, we get the height, width and depth of the image. Then atomic force microscopy that we uses a metal and diamond probe inserted into the specimen to produce 3D images and which is used for distinguishing parts of the cells. In the 3D image preparation, we use different methods. First one is photogrammetry and second one is photometric stereo. Photogrammetry means where 2 or 3 images are there from the titled specimen. And in case of photometric stereo, where there is use of 4 images from the detector. Here are some example of 3D images. First one is transparent eyeball 3D image and second one is the whole body of cancer 3D image. And the third one is fruit fly brain 3D image. So next, negative staining. So negative staining is a staining method. There are two kinds of staining, positive staining and negative staining. So staining way we get the visual of image in a colored manner, that is way clear picture of image we get through the electron microscope. So negative staining method we use in transmission electron microscope. Here it is different from the positive staining, that is the staining where we get a transparent color of specimen, but the background got the stained. But in case of positive staining, the specimen gets stained and background get a transparent view. Because here in case of negative staining, there is a repulsion because of their negative charges in between the specimen and the stain. That is why background got stained and the specimen get transparent. So this negative staining method is developed by Mark Adrian, and the negative staining refers to the practice of applying a heavy metal salt stain to a sample for observation. The stain enhances contrast and allows for initial visualization of specimen. It is based upon the principle that there is no reaction between stain and specimen, and the background is stained and leaving the actual specimen untouched and thus visible. Here are some examples of negative staining of bacteria bacilli, and the second one is about of spirillum, and of third cocci. Then disadvantages and advantages of negative staining. Disadvantages are negative staining does not provide much information about the cell rather than cell size, cell shape and arrangement. And the second disadvantage is by using this technique, we cannot examine a particular stain or a type of organism. And the advantages of negative staining is that can be performed at room temperature and provides details about the stability and homogeneity of sample and generates better contrast. And then we have to discuss about single particle reconstruction. Single particle reconstruction, here we take averages of copies of image from the electron microscope of a specimen, and this averages of the image taken, and that is why creating the 3D image of the specimen. So is the method used in 3D EM when multiple copies of the particle of interest are present. In single particle reconstruction, a 3D reconstruction of particle is generated by averaging the images of single particle produced by an electron microscope. Here sample must be pure or homogeneous. The sample is spread uniformly across EM grid in the form of a thin film and plunge frozen to embed the particle in vitreous ice or stained with a negative stain to enhance the contrast. So, some steps involved in 3D single particle reconstruction. Here we can a pictorial representation of the steps. First one, the protein purification. The purified protein be used in cryo-EM, subform the collection and then aligned and average the frames. Then defocus the determination and CTF correction. Then particle alignment and classification, and we get the final structure. Or other way, we negative staining and initial model we get, then initial model reprojection, then orientation, refinement, and then particle alignment and classification, then final structure we get. So, sample preparation, imagine electron microscope, data quality assessment, CTF determination, particle selection, 2D classification, 3D classification, 3D reconstruction, model building. These are the steps involved in single particle reconstruction. So advantages, there is no crystallization required and a small amount of sample is only required for this creation of a 3D image, and no special kind of buffer is needed for this technique, and heterogeneous samples can be used for the 3D image in single particle reconstruction. And this single particle reconstruction is a automated process and we get a high resolution way of copy. Then disadvantages, the radiation may get damages the cells of the specimen, and number of particles required, and alignment precision, and also the conformational heterogeneity and orientation preference are the disadvantages of single particle reconstruction. So thank you.